Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And in this particular module, we are discussing about the different aspects of the immunology. So far what we have discussed about the basics of immunology, the purpose of the immune system, how the immune system is having a different types of systems. So we have the cell mediated immunity, we have the antibodies to protect us and so on, right? So we have a different types of cells. We have the T cells, B cells, we have the macrophages, and how these cells are integrated to each other, and they are actually causing the very robust immune response so that we can be get protected from the different types of infectious organisms. Not only the external infectious agents, we also get immune system is also protecting us from the internal antigens as well, right? For example, it is protecting us from the development of the cancers or eliciting the different types of allergic reactions. Subsequent to that, in the previous lecture, we have also discussed about the antibodies. So we have discussed about the structure of the antibodies, how the antibody is being produced within the host system, and how the diversity of the antibody is being governed by the clonal propagation and the selection by the antigen. And then subsequent to that, we have also discussed about how you can be able to produce the antibody into animal. So we have taken an example of a rabbit and we have shown you all the steps how the antibody can be produced in rabbits. Uh, Subsequent to that, we have also seen the interaction of the antibody and antigens, and we have also studied how you can be use this particular phenomena for developing the different types of immune assays and how you can be able to use this for for different types of biological applications. Now, in this today's lecture, we are going to discuss about the vaccinations and how the vaccination is helping the organism to acquire the resistance against the different types of infectious organisms and how that can be used or how that is helping the society to get protected from the different types of diseases. So as far as the vaccine is concerned, the vaccine is also called as immunization. So immunization is the process of eliciting a long-term state of protection, immunity against a disease-causing pathogens. Exposure to the live pathogen followed by recovery is one route to immunization. Vaccination or the intentional exposure to form a pathogen that does not disease is a vaccine actually, or is another way of doing the mimicking the natural way, right? So in the naturally what happens when the pathogen comes, it actually elicited the immune response and then our immune system is eliminating that pathogen. And in this process, some amount of B cells or some amount of memory B cells are being formed. So when the same pathogen comes to the next time, it is actually eliciting the more robust immune response so that that particular pathogen is being eliminated. So the similar natural process can be mimicked if you are actually going to give the infection of the form of a pathogen that does not cause disease, right? So in ideal world, both engage the antigen specific lymphocytes and results into a generation of memory cells providing the long term protection. However, vaccination does not ensure immunity and a state of immune protection can be achieved by means other than the infection or the vaccination. For example, the transfer of antibody from the mother to fetus or the injection of an antiserum against a pathogen, both are providing the immune protection, but these are called as the passive vaccinations or the passive immunity. Without the development of memory B cells or T cells specific to the organism, however, this state of immunity is only temporary. Thus, vaccination is an event where the immunization, the development of a protective memory response, is a potential outcome of that particular event. So, this can be achieved by multiple ways, right? So, you can actually have the different types of vaccines. You can have the passive vaccines, right? So Edward Jenner and Louis Pasteur are recognized as the pioneer of vaccinations or the early attempt to induce the active immunity. But similar recognition is due to the Emil von Behring and the Hindersburg Getasso for their contribution to the passive immunity. These investigators were the first to show that the immunity elicited in one animal can be transferred to another by injecting the serum from the first. So, passive immunization, in which the preformed antibodies are transferred to a recipient, occurs naturally when the maternal IgG crosses the placenta to the developing fetus. Similarly, the maternal antibody to the diphtheria, tetanus, streptococci, rubella, mumps, and the poliovirus are afford passively required protection to the developing fetus. Later, the maternal antibody present in breast milk can also provide the passive immunity to the infant in the form of the maternal produced IgA. Then we have the passive immunizations, which can also be achieved by injecting a recipient with the preformed antibody, which are called as antiserum, from the immune individual. Before vaccines and the antibody become available, the passive immunization was the only effective therapy for some otherwise fatal diseases such as diphtheria, providing the much needed humoral defense. Currently, several conditions still warrant the use of passive immunizations, including the following. Like, for example, if there will be immunodeficiency, toxins on the venous exposure, or the exposure to the pathogen that can cause a disease faster than an effective immune response can develop. Babies born with the congenital immune deficiencies are frequently treated with the passive vaccinations, as the children experiencing acute respiratory failure caused by the streptococcal virus. Passive immunity is used in an unvaccinated individual exposed to the organism that cause the botulism, tetanus, diphtheria, hepatitis, measles, and the rabies, or to protect the travelers and the healthcare workers who expect exposure to the potential pathogen for their lack of protective immunity. Anti-serum provide the protection against the poisonous snake and the insect bite. In all these instances, it is important to remember that the passive immunization does not activate the host immune response, right? So, passive immunization is only providing the solution. Passive vaccine immunization does not provide the protection for the subsequent infections, right? Passive immunization is a temporary solution. Therefore, it generates no memory response, and protection is transient. Protection is temporary. It actually happens at that moment. You cannot produce the antibodies, so you are taking the antibody from the somebody else, right? So, passive immunization may be effective. It should be used with caution because certain risks are associated with the injection of a preformed antibody. The antibodies were produced in another species, such as horse, right? The recipient can mount a strong response to the isotypic determinants of the foreign antibody or the part of the antibody that are unique to the horse species. This anti-isotype response can cause serious complications. Some individuals will produce the IgE antibody specific for the horse specific determinants. High level of these IgE horse antibody immune complexes can induce the perceived mast cell degranulations, leading to the systemic anaphylaxis. Another individual produces IgG or IgM antibody specific for the foreign antibody, which forms complement activating immune complexes. The deposition of these complexes in the tissue can lead to the type 3 hypersensitivity reaction. Even the human antiserum or the gamma globulin is used, the recipient can generate the anti-allotype response to the human immunoglobulins and the recognition of within specific antigenic differences, although its intensity is usually much less than that of anti-isotype responses. There are common agents which can be used for the passive immunization. Like, for example, the black widow spider mite, you can actually use the horse antivenom. Then botulism, you can use the horse antitoxins. Bacteria, you can use the horse antitoxins. Then for the hepatitis A and B, measles, tetanus, you can use the pooled human immunoglobulins. Then for the rabies, you can use the human or horse polyclonal antibodies. Then for the snake bite, you can use the horse antivenom, and the respiratory diseases, you can use the monoclonal anti-RSV. Then we have the active vaccinations or the active vaccine. Whereas the aim of the passive vaccination is transient, the protection or the alleviation of an existing condition, the goal of active immunization is to trigger the adaptive immune response in a way that will elicit the protective immunity and the immunological memory. When active immunization is successful, a subsequent exposure to the pathogenic agent elicits a secondary immune response that successfully eliminates the pathogen or prevent the disease mediated by its product. Active immunization can be achieved by the natural infection with a microorganism, or it can be acquired artificially by administration of a vaccine. In active immunization, as the name implies, the immune system plays an active role, right? Proliferation of the antigen specific T cells and B cell is induced and results into a formation of the protective memory cell, and this is a primary goal of vaccination. So active vaccination is actually going to be engaged the T cell, B cell, and ultimately it is actually going to produce the memory cell, right? So these memory cells are actually going to have the memory for a particular antigen, right? And when this antigen comes naturally, right, when you are going to get a disease, then these memories are actually going to produce the antibodies, and that's how these antibodies are actually going to protect you against the particular disease. The active immunization with various types of vaccines has played an important role in reduction of death from the infectious diseases, especially among the children. The use of vaccines for active immunization has proven essential for lowering the number of infectious disease related fatalities among children. According to the recommendation established by the American Academy of Pediatrics, right, pediatric vaccination in the United States starts from the birth, right? So you know that there are so many vaccines which we are going to put or we are going to give to the kids, right? And it starts from the day one, right? So in India, when the kid is born, the day one you are actually going to get the vaccine for the BCG, right? And BCG vaccine is being used for protect the person from the one of the deadly disease which is called as the tuberculosis. So for the hepatitis B vaccine, diphtheria vaccines, pertussis vaccines, hemophilia influenza type vaccines, inactivated polio vaccines, measles, mumps, rubella vaccines, varicella zoster vaccines, meningococcal vaccines, etc. are among the vaccines which are advised for the children up to the class age 6. In addition to girls, boys between the age of 11 to 12 are advised to get vaccine against the HPV or human papilloma virus, which is actually going to be used for generating the immune response against the cervical cancer, right? So it is recommended not only for the girls but also for the boys. Then we have the subunit vaccines. So many of the risks associated with the attenuated or killed whole organism vaccine can be avoided with a strategy that uses only the specific purified micromolecule derived from the pathogen. So subunit vaccine is part of the pathogen, right, or part of the pathogen in terms of the protein, right? So you are not using the complete pathogen, but you are using a protein from the particular pathogenic organism. Three most common application of this strategy referring to a subunit vaccines are inactivated exotoxins or toxoids, capsular polysaccharides, or the surface glycoprotein, and they are and key recombinant protein antigens. One limitation of some subunit vaccines is, especially the polysaccharide vaccine, is their inability to activate the T helper cells. Instead, they activate the B cell in a thymus independent manner, resulting into a IgM production but little class switching. So no affinity maturation and little, if any, development of memory cells. So this is the limitation of some of the subunit vaccines where you are using the polysaccharides or the low molecular weight antigens, that it is not going to activate the T cells and it is not going to activate the B cell in a thymus dependent process, and because of that it is actually going to produce the IgM rather than the IgG, and it is also not going to give you the memory cells. However, the vaccines that conjugated a polysaccharide antigen to a protein carrier can take care of this and inducing the T helper cell response. Some bacterial pathogens, including those caused diphtheria and tetanus, causes the exotoxin that account for all or most of the disease symptoms. Diphtheria and tetanus vaccines have been made by purifying the bacterial exotoxins and then inactivating it with the formaldehyde to form a toxoid. Vaccination with the toxoid induces the antitoxoid antibodies, which are capable of binding to the toxin and neutralizing its effects. Conditions for the production of toxoid vaccines must be closely controlled and balanced to avoid the excessive modification of the epitope structure while also accomplish the complete detoxification. As discussed previously, the passive immunity can also be used to provide the temporary protection in the unvaccinated individual exposed to the organism that produce these exotoxins, although no long-term protection is achieved in this case. The virulence of some pathogenic bacteria depends primarily on the anti-phagocytic property of their hydrolytic polysaccharide capsules. Coating the capsule with antibody can complement the greatly increase the ability of macrophage or neutrophils to phagocytize such pathogens. These findings provide the rationale for vaccine consisting of the purified capsular polysaccharide. There is a vaccine for the Streptococcus pneumoniae, right? And that contains the 13 antigenic distinct capsular polysaccharides. The vaccine induced the formation of opsonizing antibody and it is now on the list of the vaccines recommended for all the infants. The vaccine for Neisseria meningitis, a common cause of bacterial meningitis, also consists of the purified capsular polysaccharides. Some viruses carry the surface glycoproteins that have been tested for use in the antiviral vaccine with a little success. Similarly, we have a glycoprotein D from the HSV2 and that is being used to prevent the genital herpes in the clinical trial of some vaccines, suggesting that they may have a variable approach for the antiviral vaccines. Theoretically, the gene encoding any immunogenic protein can be cloned and expressed in a cultured cell containing recombinant DNA technology, and this technique has been employed widely in the design of many types of subunit vaccine as well. For example, the safety way to produce a significant amount of purified toxin to go into a generation of the toxoid vaccines, cloning the exotoxin gene from the pathogenic organism into a easily cultured host cell. A number of genes encoding the surface antigen from the virus, bacteria, and proteozoan pathogens have been successfully cloned into a cellular expression system for use into a vaccine development. Like for example, the recombinant vaccine developed against the hepatitis B vaccines developed by cloning the gene to a major hepatitis B surface antigen and expressing it in the yeast cells. The recombinant yeast cells are grown in large ferments, allowing the HbA to accumulate into the cell. The yeast cells are harvested and disrupted, releasing the recombinant HbA antigen, which is then purified to our conventional biochemical techniques. And the recombinant hepatitis B vaccine induced the production of protective antibodies and holds much worldwide promises for protection against the human pathogens. So this is the protection of the subunit vaccines like the hepatitis B vaccine. So this is a hepatitis virus, right? So from the virus, what you do is you take out the coat protein, right? And then you coat protein, you clone it into a suitable yeast vector, and then you transform that into a yeast, and then it is actually going to start producing the protein, and then you use the purification strategies, and that's how it is actually going to be a vaccine. Then we have a DNA vaccines, so a more recent vaccination strategy called a DNA vaccines utilizes the plasmid DNA encoding the antigenic protein that are injected directly into the muscles of the recipient. This strategy relies on the host cells to take up the DNA and produce the immunogenic protein in vivo, thus directing the antigen through endogenous MHc class 1 presentation pathway, is helping to activate the better CTL responses. The DNA appears either to integrate into the chromosomal DNA or by maintained for the long period in an episomal form, and it will be taken up by the dendritic cells or the muscle cell into the injected area. Since muscle cells express low level of class 1 MHc and do not express co-stimulatory molecule, delivery to local dendritic cell may be crucial for the development of antigenic responses to the DNA vaccine. So far in the subunit vaccines, what we are doing, we are cloning that part of that particular pathogen into a recombinant vector, and then we are producing the protein, and then we are injecting the protein, right? In a DNA vaccine, instead of doing that, we are actually taking out the gene fragment, cloning it into a suitable vector, and then we are injecting the vector, right, containing that particular recombinant DNA. And once this particular recombinant clone enters into the cell, it starts producing the protein. And then as a result of that, it is actually going to start, eliciting the immune response, and that's how it is actually going to mimic the natural conditions, right? So tests in the animal have shown that the DNA vaccines are being able to induce the protective immunity against a number of pathogens, including the influenza and the rabies. The addition of a follow-up booster shot with the protein antigen or inclusion of a supplementary DNA sequence in the vector may enhance the immune response. One sequence that has been able to added to some vaccine in the common CpG DNA motif found in the some pathogen, recall it is sequence in the ligand for TLR9. DNA vaccines offer some potential advantage over many of the existing vaccine approaches. Since the encoded protein is expressed in the host in its natural form, there is no denaturation or the modification. The immune system is directed to the antigen exactly as it is expressed by a pathogen, inducing both humoral and the cell-mediated immunity. To stimulate both the arms of the adaptive immune response, non-DNA vaccines normally require immunization with a live attenuated preparations, which incur additional risk. So DNA vaccines also induce the prolonged expression of the antigen, enhancing the induction of the immunological memory. Finally, DNA vaccine presents an important practical advantage, right? There is no refrigeration of the plasmid DNA is required, eliminating the long-term storage challenges. Remember that vaccine storage and transport is a very important feature, right? Because it actually eats up a lot of money also, right? So when you are supposed to store the vaccines at a very low temperature, then you are supposed to carry the refrigerators and all that, and that actually increases the price of these vaccines. So in addition, the same plasmid vector can be custom tailored to insert the DNA encoding a variety of protein, which allows the simultaneously manufacturing of a variety of DNA vaccines for the different pathogens, saving the time and the money. An improved method for administering the DNA vaccine inhale like coating gold microbeads with the plasmid DNA and delivery of the coated particle with the skin into the underlining muscle with an air gun, which is called as gene gun, right? This allows the rapid delivery of a vaccine to a large population without the need for a huge number of needles and syringes, improving with the safety and the cost. Human trials are underway with the several different DNA vaccines, like including those for malaria, HIV, influenza, Ebola, and herpes. Although there are currently no licensed human DNA vaccines, the three such vaccines has been licensed for veterinary use, including West Nile vaccine that is been protecting in horses. This vaccine has been tested in humans. After 3 doses, most volunteers demonstrated the titer of neutralizing antibodies similar to those seen in horses, as well as CD8 positive and CD4 T cell responses against the viruses. Since the widespread development of DNA vaccine for use in humans is still in its early stage, the risks associated with the use of this strategy are still largely unknown. So this is the classical, the strategy for the DNA vaccine. So first what you are going to do is you are going to take out the gene, right, what you are going to clone, and then you are going to clone that into the vector. Then you are going to insert that vector into a particular cell, right? Then you inject this, right? And then it is actually going to start producing the different types of vaccines, right? And then you can actually be able to use the DNA vaccine. Similarly, we can have the RNA vaccines. So RNA vaccines, by priming the body to combat the external invaders, the vaccines aid in the prevention of infection. So all vaccines cause an immune response by introducing a harmless fragment of a specific virus or bacteria. The majority of vaccines contain a dead or the weakened virus or the bacteria. But instead of using a component of a real bacteria or viruses, researchers have created a new kind of vaccine that is being known as the messenger RNA vaccine. So one kind of RNA that is required for the synthesis of protein is messenger RNA, right? Cells strictly degrade the messenger RNA after completing the protein, right? So vaccine derived messenger RNA do not change DNA or enter the nucleus. So a fragment of RNA that matches a viral protein, typically a little fragment of a protein present on the virus outer membrane, is introduced as a part of messenger RNA, right? So earlier we were putting the DNA vaccine. So DNA is producing the messenger RNA, right? And then messenger RNA is producing the protein, right? Remember that when we were discussing about the central dogma of molecular biology. Now, with the DNA vaccine, there is a danger that DNA vaccine could integrate into the the human genome or the organism genome. So to avoid that, people have started injecting the messenger RNA. Now, messenger RNA will not integrate into the genomic DNA or genomic content, right? So messenger RNA will produce the protein, and then the protein is going to be used as the antigen, and it is actually going to elicit the immune response. So those who receive a messenger RNA vaccine are not exposed to the virus, nor can be vaccine cause the infection. The viral protein can be produced by simply using the messenger RNA. The immune system creates a specialized protein known as antibody in reaction to the protein's recognition as a foreign. By identifying the certain viruses or other pathogens, binding to them and designing the pathogens for eliminations, antibody aid in the body's defense against the infection. Once created, the antibody stayed in the body even after the pathogen has been eliminated, allowing the immune system to react promptly in the event of re-exposure. After being vaccinated against the virus using the messenger RNA, antibody may swiftly identify the virus, bind it, and label it for the destruction before it might cause the major sickness. The only recognized or permitted messenger RNA vaccine at this time are for the COVID-19, a illness brought by the SARS-CoV coronavirus. These vaccines employ the messenger RNA, which instructs the cells to make the copies of the spike protein, a protein found on the surface of the coronavirus. Scientists are looking into the possibility of using the messenger RNA to create the vaccines for the adult illness. This is the the examples of the COVID virus. So this is the COVID SARS-CoV virus, right, which is responsible for the famous COVID-19. So the SARS-CoV virus, which was initially discovered in China in late 2019, is the cause of the viral disease, which is called as COVID-19. When an infected person coughs, sneezes, talks, or breathes, respiratory droplets are the main way of the virus spreads, and then it causes the different types of illness and the symptoms, right? And talking about the COVID vaccine, the COVID virus is a primary target for the vaccination because of its spike protein, which enables it to bind to and penetrate the human cells. The COVID vaccines are being designed to train the immune system to recognize and fight the SARS-CoV, preventing illness or reducing the severity of the infections. The spike protein or the whole viruses, right? That these are the two things with what can use for developing the COVID vaccine. Now talking about the messenger RNA vaccines, so messenger RNA vaccine introduced the cells to how to produce the spike protein, and that is presented on the COVID-19 virus's surface. So following the immunization, the protein fragments are produced by the body's muscle cell and display on their cell surface. The body produces the antibody as a result. These antibodies are then employed aid in the removal of COVID-19 virus if they acquire it. Once the protein fragments are formed, the instructions are being broken down and eliminated by our cells. The vaccine's messenger RNA does not penetrate the cell's muscle, which house the DNA. Messenger RNA is used in the Moderna and the Pfizer's Bionet COVID-19 vaccine. So in a COVID-19 vaccine, you are actually producing from the COVID-19, you are making the messenger RNA, right, which are responsible for making the spike protein. And then you are putting this messenger RNA into a human cell, right? And then this human cell is started producing the protein, and it is actually expressing that protein, spike protein, on their surface. And because it is immunogenic, it is actually going to be recognized by the immune system, and it is actually going to start producing the antibody. And now when there will be a subsequent infection with the real virus, then these antibodies are actually going to bind these antibodies, these viruses, and they will not allow the virus to cause the disease. Then we have the vector vaccines. So this is a kind of a vaccine involving the inserting component from the COVID-19 virus into a modified trait of another virus, with this referred to as a viral vectors. So cells are instructed to replicate the COVID-19 S protein by a viral vector. The immune system reacts by producing the antibody and protecting white blood cells when the cell exhibit the S protein on their surface. So the antibody aid in the removal of COVID-19 viruses. So neither the viral vector nor the COVID-19 virus can infect a person who has received the viral vector disease. So for example, the Johnson & Johnson COVID-19 vaccine. So this is the strategy for developing the COVID-19 vaccines. So you are actually going to have the COVID-19 virus. You just take out the spike gene for the spike proteins. Then the clone it into the viral vectors, and then you are going to insert that into the human cell, and then it is actually going to start producing the spike proteins. And then these spike proteins are going to be recognized by the immune system, and then it is actually going to produce the antibodies, and these antibodies can be used in a subsequent infection to provide the immunization. Then we also have the protein subunit vaccines. So only the portion of a protein that causes the effectively activate the immune system and included in the subunit vaccines, right? So you're basically going to take the spike protein and then you produce the spike protein and then you are going to use that. So examples of the subunit vaccine is Novavax COVID-19 vaccines. And this is the strategy, right, where you are actually going to take out the spike proteins, clone it into a bacterial or the yeast or the animal system, then you produce the protein, then you purify that protein, and then you inject that protein into for vaccinations, and then the spike proteins are going to be recognized by the immune system, it will produce the antibodies, and then these antibodies are going to give you the protection. Then in some cases, we have used the inactivated COVID vaccine, right, or inactivated COVID virus and as a vaccine, right, or complete virus that has been killed so that it cannot reproduce or spread illness is being used in the inactivated vaccines. Example is the Covaxin, which is being developed by the Bharat Biotech, right, and this is being majorly being used for vaccination in India, right. So what you have is this is the virus which causes a disease. So what you can do is you culture this virus and then you inactivate this virus by chemical treatment or some other method. Then you take this and inject it into the human, and then the humans are going to produce the antibody and they are actually going to give you the protection. Then we can also have the DNA vaccines. So a small fragment of DNA known as plasmid is introduced into the body in a DNA vaccine. And what you can do is you just take the gene for the spike protein and then you are actually going to encoded that into a vector, right, and then you inject that as a DNA vaccine. So the examples of DNA vaccine is ZyCoV-D, the first authorized DNA based COVID-19 vaccine which is produced by the Zydus Cadila in India. And this is the classical examples, right? So you're going to have a COVID-19, you are going to take out a portion of the chromosome which is responsible for generating the spike protein. Then you clone that into a suitable viral vector and then you inject that into the cell and they will start producing the proteins, and these proteins are immunogenic, so they are actually going to be recognized by the immune system, and that's how they are actually going to produce the antibodies, and that's how these antibodies are going to give you the protection when there will be a real infection of the COVID-19. So this is all about the vaccines. What we have discussed about the basic phenomena, what is happening when you are getting the vaccinations. So when you are getting a vaccination, the purpose of the vaccine is that it should actually elicit the immune response, but it will not cause a disease. And as a result of that, it is actually going to train the immune system, and it is also going to produce the memory cells, right? And these memory cells are actually going to keep a memory, and as a result of that, you are actually going to, whenever there will be a subsequent encounter with the real organisms, it is actually going to elicit the immune response, and that's how it is actually going to clear the infection. As far as the purpose is concerned, you can actually have the different types of vaccines. You can have the passive vaccinations, you can have the active vaccinations, and within the active vaccinations you can actually use the different strategies. So in some cases people are using the inactivated organisms, and in some cases people are using the product from the the product from the pathogens, such as toxides or toxins, which will not cause the disease, but it is actually going to train the system so that you can actually be able to elicit the immune response. Then we can also use the subunit vaccines, you can actually use the DNA vaccines, you can use the messenger RNA vaccines, and so on. And lastly, we have also discussed about the different types of vaccines which are being developed against the COVID-19, and we have also understand the mechanism by which these vaccines are being developed and how they are actually giving the protection against this deadly disease. So with this, I would like to conclude my lecture here. In our subsequent lecture, we are going to discuss some more interesting aspects. Thank you.